Hello student, welcome to Department of Zoology, Sri Mulika Devi Mahavidyalaya Nigoz, Taluka Parner, District Ahmednagar. In previous video lecture, we was studied some important disease. For example, in bacterial disease, that is muscardine and types of muscardine, then control measure, then symptoms of muscardine. This point is already studied in previous video lecture. Now today I will discuss the remaining diseases of silkworm. So let us start to study the diseases of silkworm. The second part of diseases, that is viral disease. These microorganisms cause 70% damage to sericulture industry. You know, in previous video lecture, I was give the information the sericulture is mainly affected by two factors, that is pest and diseases of silkworm, because if any type of pest and disease attack on silkworm, that time you are lost one single cocoon from the in the form of one single larva. So if you can lose the single cocoon, it is directly affected your final production of silk. So let us study some viral diseases of silkworm. In that first disease named as nuclear polyhedrosis (NPV). This disease is known as jaundice in France. In France, it is very popular jaundice disease named as nuclear polyhedrosis. And that it is also called as milky disease, fatty degradation disease, and hanging disease. These are the symptoms, some symptoms of polyhydrolysis. So the farmers or researcher give the name, for example, jaundice disease in France, milky disease, fatty degradation, and hanging disease. These all the names are directly belong to the this disease, that is nuclear polyhydrolysis disease. This is viral disease of silkworm. This is very serious disease occurred in throughout the air. In India, particularly in Karnataka, it is called as halahula disease in local language. Infection. The disease is mainly caused by Borrelia bombycea. These are the name of virus. If the infection mainly through the mouth after the infection of virus, means if your mother silkworm is infected by this virus, this infected mother can be transfer or spread this disease into the whole colony or inside the rearing tray through the mouth. The disease occurred with the digestion of polyhedra in the alimentary canal of the silkworm larva. You know why you can call as polyhydrolysis? Because the digestion of polyhedra in the alimentary canals, they are directly belong to the name of disease. If disease cause the digestion of polyhedra in the alimentary canal of silkworm, this virus multiplies in the form of polyhedra in the nucleus of the tracheal epithelium cells, then adipose cell, then tissue cell, then dermal cells, and blood cell of silkworm. Means they are show variation in their multiplication. If your silkworm is infected with this virus, they are easily survive or easily get the serious infection through the mode of multiplication. Symptoms of this virus. The disease cannot be identified during the earlier stage of infection because this disease is mainly target to the digestion of polyhedra inside the alimentary canal. So farmers or sericulturist is not easily identified the disease infection is occurred in rearing trend because your alimentary canal is internal part of the silkworm, so you are not easily easily analysis of your alimentary canal of silkworm, except the sluggish nature is found in your silkworm. Initially, the skin show oily and shiny appearance. The secretion of oily substance, that time they are show shiny natures on the dorsal surface of silkworm. This is one of the initial key of identification. In advanced stage, larva loss appetite, that is, they stop the feeding or stop the feeding, skin become thin, fragile, the body become milky white with intersegmental swelling. Means if your silkworm was affected by this virus, that time the larva firstly stop the feeding of mulberry leaves, then skin become a thin, very thin skin is present in your larva. Then last identification, that is body become a milky white with intersegmental swelling, means whatever the segmentation is present in your body, if you are finding intersegmental swelling inside the larva, these are the segmental. If in here your larva is show swelling, that time you are easily understand your larva become affected with nuclear polyhedrosis, that is NPV disease. I hope you are understand what is the symptoms, what is the mode of infection in NPV disease, that is polyhydrolysis disease, nuclear polyhydrolysis disease. Now, last point of this disease, that is how to control. The controlled silkworm rearing under the hygienic condition. It is very important. Every time I was told you, if you want to perform the sericulture, that time it is very important to maintain the hygienic condition inside the rearing tray and as well as inside the rearing house. Disinfecting the rearing rooms, then appliances, mulberry storage rooms, molting rooms, rearing permission are followed by the using this all the or applying this all the precautions to avoid the infection of NPV disease. Further, avoiding feeding unsuitable leaves. That is very important. Unsuitable leaves means if your mulberry leaves is very matured, unhygienic, or they show some dusty natures on their appearance, that time you can avoid these leaves for the feeding. Proper ventilation and spacing, timely picking out and destroying the diseased worm are some of the precautionary measures against the NPV disease. You know, proper ventilation, it is very important during the sericulture. If the crowded condition present in rearing tray, that time they are communicate or body contamination is easily happens. Secondly, spacing is very important because to give the proper space, to give the proper ventilation means air pass easily, then timely pick up out, destroyed the disease worm. If any type of the diseased worm is found in your rearing tray, you can pick up and destroy it outside the rearing tray or rearing house. It is a precautionary measures against the any pest and disease infection. The diseased worm contain number of fresh polyhedra. Thus, the worms are removed from carefully without breaking the skin. You know, in this disease, the skin of larva is very thin. So if you can find out any disease infected disease infected larva, and that time if you want to pick up out from the rearing tray, but it is very skillful work. So to uh, carefully without breaking the skin, because all the body is contaminated with polyhedra. So if the polyhedra is spread on rearing tray, it is chances increase the chances to increase the contamination. So breaking is easily possible because skin is very thin. So avoid this problem and to remove out your infected larva from the rearing tray and burn it or destroy it outside the rearing house. The deceased worm or dead larva are disposed carefully by putting in limp post or by burning. This point is already discussed within this slide, because if any type of the larva find out with the infection of this disease, it is first necessary steps to pick up and to kill or to burn this infected disease from the outside of the rearing tray. Extremely low and high temperature and humidity during the rearing should be avoided. This is very important, because if any type of the any type of unsuitable condition, that is very low temperature, very high temperature, very high humidity, very low humidity, these are totally avoided during the rearing techniques or rearing of silkworm, because this condition is offered to contamination of disease, or it will be increase the chances of to develop the disease inside the rearing house. So you can avoid this condition in your rearing house. I hope you are understand this disease, that is NPV disease. In that I will give the information regarding what is nuclear polyhydrolysis disease. It is called as jaundice disease in France, milky disease, fatty degradation disease, and hanging disease. In Karnataka, it is called as hula hula disease. The bacteria, sorry, virus name as Boronilla bombaceae. It is a virus name they cause this disease. Then the area or organs particularly affected by this disease is polyhedra in the alimentary canal. So you are not easily identified in initial stage, but if serious infection of this disease, you can find out some symptoms, that is loss of appetite, the larva stop the feeding, then skin of the larva is very thin, fragile, then body become a milky white, and swelling in intersegmental portion. You can easily identify this disease. Then control measure to try to maintain hygienic condition, disinfectified all the appliances and rearing house. Then to avoid the unsuitable leaves for the feeding, then provide the proper ventilation, proper space inside the rearing tray, and destroyed the any disease infected larva from out destroy the disease infected larva immediately if you are finding inside the rearing tray. Then last precaution, that is it is very important to avoid a very high temperature inside the rearing tray and very low humidity inside the rearing house. This condition is suitable for the grown up of disease, so you can avoid and to protect your larva from the infection of nuclear polyhydrolysis disease. I hope you are understand what is the uh, symptoms, then control measure, and uh, basic information regarding this disease. Now let us start to study the second viral disease named as cytoplasmic polyhedrosis. This disease is mainly found in summer season. Infection. The disease is caused by Smithia virus. Infection takes place through the polyhedra. Again, show the same nature of infection, that is polyhedra contaminated with mulberry leaf. Now here I was give the information regarding suitable leaves. These contaminated leaves, it is the biggest reason to cause this cytoplasmic polyhedrosis. So it is very important to provide the fresh and clean leaves to the your larva. Infection in the rearing tray is more common since polyhedra is released in excreta of silkworm. You know, if any larva is contaminated or infected by the cytoplasmic polyhedrosis, and by chance they are start the they start the excreting their waste product from the body, and this excreta contain the polyhedra, fresh polyhedra. So the contamination is vastly spread inside the your rearing tray. Symptoms. The infected larvae show slow growth, stunted body, reduce the mulberry consumption, that is appetite, and look dull white in color. The color of larva, it is pure whitish in color. But here you are observe some downness or lighty white colored if your larva infected by the this disease. Unequal size worms are common and the worms show delayed molting. The if your larva is not matured or not show well growth, that time you are observed these symptoms. Then if your larva is not complete their growth, that is they show the final delay in molting. Larva loss appetite, that is they stop the feeding. Then legs behind the normal larva in their development. If infected larva, grown up larva, the thorax become transparent. If infected larva and your normal larva, they show the difference, that is, their thorax become transparent. Means the whiteness is very dull. Control, again they show the same control measures, that is disinfection of rearing room, appliances used in during the rearing of silkworm, and surrounding area should be carried out using the 2% formalin solution and 5 0.5% fresh slack lime powder. Using this chemical, you can disinfectify all the area of rearing rooms. Second control, that is dead larva, infected larva, excreta of particular infected larva, unused leaves or reused leaf should be destroyed by burning outside the rearing house. These are the precaution measures against this viral disease. It is very important because if you can avoid the infection of cytoplasmic polyhydrolysis, it is very important to try to maintain hygienic condition inside the rearing tray and rearing house. I hope you are understand what is the symptoms and what is the control measures of polyhydra and cytohydrolysis poly diseases. Now, next one, that is silkworm flacheries. The term refers to feckled condition of silkworm larva suffering from the dysentery. Dysentery is happened in this condition, that is called as flachery. Flachery is subchlorine resulting primarily due to physiological disorder and secondarily by the infection of bacteria. If your silkworm infected with any bacteria, they show dysentery or vomiting. These are the physiological disorder observed in silkworm. In that first one, that is viral flacherie. Flacherie, it is two types, that is bacterial and viral. In between, they are show some common symptoms. Flacherie is most common, caused by high humidity and fluctuation of temperature inside the rearing rooms. If you are not maintain the temperature or humidity, bacteria and viruses attack on your silkworm and they are easily infected by the flacherie. So you can try to maintain the optimum temperature and humidity inside the rearing tray. Worms appears lethargic and flaccid growth is stunted. Means if any type of the bacteria and viruses infected by your larva, the growth is not well in condition. You know, it is a natural process. If your health is not good, that time you are not develop or not show the well growth in silkworm. The cephalothoracic region may be transcoolant. Dead worms purified quickly, often a foul odor. You know, if your silkworm is infected by this disease, so they starting the vomiting and dysentery, and these all both the condition is produce some foul odor, that is bad smell. It is called as a bad smell. So another healthy silkworm start the irritation, then they're not feeling good inside the rearing room, so they become restless. So again, this condition is not favorable for the healthy larva also. So it is your duty to remove, to transplant, to remove the dead larva, then dysentery material, excreta, vomiting of the silkworm immediately from the rearing tray and discarded outside the rearing house. How to control? A flachery is most often disease in the earlier instar and does not present until the later. You know, during the younger period, this disease is very common in all the silkworm. In star, it is usually impossible to eradicate without destroying the entire colony. Means it is your duty to provide the hygienic condition and good quality of food material to your larva in earlier stage, that is younger stage of larva, so avoid to this contamination of diseases. Remove the stuck and dead silkworm and excreta, then another material, waste material secreted or secreted by larva. It is very important to remove immediately from the rearing tree. I hope you are understood all the points, all the diseases of today's lecture. If you have any question, any difficulty regarding this two disease, that is first two is belong to the viral disease and third one it is a combined disease that is called as silkworm flacherie, both in that is viral and bacterial flacheries. If you have any questions regarding this point, you can ask me in comment box. Thank you very much to all of you.